let me put it this way. So I wanted to mention first, when we do preclinical, that we sometimes use different models to study new targets. Now we discovered that there is something out there that's called the microenvironment. And the microenvironment may affect how we see the targets in the cells. And these are examples that we published last year, in which we put lymphoma cells, B-cell lymphoma cells, into a three-dimensional, oops, it's going, hold on. Now, into a three-dimensional matrix that contains the stroma cells and also extracellular matrices. And the response of these cells to common drugs like doxorubicin and panovinostat change if you compare when they're in the 2D regular culture versus a 3D. So now something that is a target is not a target anymore. And the same happens when you put, for example, these ABC, DLBCLs that in the 2D system, they don't have a target for ROCK inhibitor, for example. But in fact, when you put in 3D, this become a new target. So if you do this with several compounds, there are more than 400 compounds, and you compare 2D versus 3D culture, the microenvironment is adding or taking out several targets. So this is telling us that when you do preclinical testing of the drug, it's very important to consider the system that you're using. And also when you read the papers, if the people is using 2D system or 3D system, because it can lead to totally different conclusions. But as well, it's telling us the microenvironment is very important regulator of the response to the drugs. For that, when we look into the new novel therapeutic targets, we look into the B-cell lymphoma cell itself, but also on the extracellular microenvironment that includes the intracellular matrix components, also cytokines, angiogenesis, and other factors, including metabolites, and the immune infiltrating cells. So all these are active research targets. These are just a few examples. Selected therapeutic actionable microenvironmental pathways. Some of these, like LENA, for example, has also activity in the B-cell lymphoma itself, but is primarily targeting the microenvironment. And obviously, there's a lot of research ongoing on PD-1 and PD-1 inhibitors. And many of the therapeutics now in the preclinic is try to understand how we can increase the effect of these immune checkpoint inhibitors. For example, okay, here is an example of a combination of LENA with PD-1 and PD-1 inhibitors published in multiple myeloma cells. Also, arginase inhibitors seem to be working towards increasing the activity of these drugs. There are many examples that other drugs increase the presentation of neoantigens in the cells that can also be combined with PD-1 or PD-1 inhibitors. So these are very areas of active research right now for us. Regarding the B-cell lymphoma cell specific targets that we are looking into, well, this is an overview of the therapeutic actionable pathways in lymphoma that we have validated over the years. So we already mentioned that the PI3 kinase, AKT, mTOR pathway are very nice target for lymphomas and mostly validated in the clinic. Obviously, the BCR receptor pathways. Down the pathway, we have the MALT, 40-MALT1 with certain drugs that are being developed for the clinical use, and as well as the TLR and MIT-ATA pathway can be targeted. All this in general result in the activation of NF-kappa B, elusive target for targeting. However, there are some drugs that can be used, for example, NIC inhibitors are recently been developed in the CD13 pathway. And there is a certain STAT3 inhibitor, also a complicated target to inhibit directly. This is an antisense oligonucleotide that's been used in the clinic that has an interaction with the activation of NF-kappa B in B-cell lymphoma. And at the core of these lymphoma cells are the three oncogenes that together with the NF-kappa B are subject of active research, BCL2, MYC, and BCL6. BCL2, well, the drugs are already in the clinic, very proven target. MYC, extremely elusive. The drug that I put here is a mix-max inhibitor. However, this cannot be used in vivo, so a still complicated target. And BCL6 is something that in our group we've been targeting for years. We have a preview version of a peptidomimetic called BPI, and I will present now some data that we published this year of an improved small molecule called FX1 for BCL6. So this small molecule is a high affinity inhibitor. It's higher than our previous version. It's called 79.6. It works through the same binding domain. It's the BTB domain of BCL6, represented here by the dimer in blue and in pink. So the drug binds to the same pocket with much higher affinity and even higher than the endogenous ligand, which is the co-repressor SMART. A consequence of this is highly potent in vivo. This is a GCV DLBCL xenograph and a head-to-head comparison with our previous version of the drug and the new version showing improving potency. Because of this, it's also active in ABC DLBCL xenograph and as well in primary patient samples from ABC tumors. So BCL6 remains a very interesting target. However, lymphoma, B-cell lymphoma, as like T-cell lymphomas, are very heterogeneous, and they are driven for more than one oncogene. So this is the situation in general with the oncogenes in B-cell lymphoma. Frequently expression of 2 or 3 of oncogenes, and on top of that, the oncogenes regulate each other. In this case, BCL6 inhibits MYC and BCL2, and they control very overlapping redundant pathways. And this is a problem sometimes when you have a very specific and very potent therapy that we prove in preclinical models, because the inhibition of BCL6, for example, using any of these drugs, will make the other 2 oncogenes to be overexpressed. So now you have a tumor, a lymphoma, which is driven by the other 2 oncogenes. They regulate pretty much the same survival pathways. So this is something that we call on-target resistance obviously in the surviving cells. This phenomenon is called oncogene switching, right? So you have a lymphoma that in general, because of the heterogeneity, driven by more than one oncogene, you kill one oncogene and the other 2 or 3 or 4 appear and they take over of the survival. So they switch from one to the other. So this is the mechanism. Here you can see in the cells, you give the BCL6 inhibitor and the BCL2 now is more prominent, either BCL2 or any of the members of the BCL2 family, like BCL-XL or MCL1. So the survival of these cells now depends on another oncogene. This obviously is an opportunity to add a new therapy to the regimen. Now you have you can add a BCL2 inhibitor to kill the remaining cells, right? Like here, represented by ABT737 or obatoclax, or you can increase the natural inhibitors of the BCL2 pathway, like using bortezomib or an 8A inhibitor. So it's a combination of different therapies, selected therapy for different, targeting different oncogenes that results in general in higher anti-lymphoma effect than each drug alone. But there's a limitation on how many oncogenes that you can target. As I showed before, there is no way to target, for example, MYC and so on. So there is we are proposing a different strategy for to avoiding this effect of oncogene switching by using or by understanding how these oncogenes are expressed. Oncogenes are not there by themselves. Any of these require to be expressed, to be present in the cell and active, a supporting network of other different proteins. I mentioned here some of these ones, but there are many others. We are very interested in HSP90, but all these, and AF4E, for example, but all these proteins support the activity simultaneously, expression of these oncogenes at the same time. So how they do this? As I said before, we're interested in HSP90, so we study how HSP90, which is a chaperone protein, not an oncogene body cells, support proteins in lymphoma, oncogenes in lymphoma. So we did an investigation of all the proteins that bound to HSP90 in lymphoma cell, and we found that all these proteins, represented here in pathway, are bound and maintained, sustaining the activity by HSP90. And remarkably, the B-cell receptor signaling. As you can see here in blue, all these proteins in the B-cell receptor signaling and downstream are maintained and sustained by HSP90, so put together, and the activity is kept by them. So we think that an idea will be to target in the oncogene supporting network to simultaneously weaken several pathways at once, and that will eventually improve the activity of oncogene-directed therapies and avoid the idea of resistant and oncogene switching. So there are several drugs that we are studying in the lab that will target all these different supporting network of proteins. I will mention just HSP90 inhibitors, PUT71, to start. So here, this is a picture that we have now after the proteomics I mentioned before. HSP90 is supporting the B-cell receptor pathway, CD40, and PI3 kinase pathways simultaneously. So if we give an HSP90 inhibitor, it's coming? Yes. Like PUT71, we think that all this, the system will shatter, will be weakened somehow. And that will be much easier then to target specifically all these pathways using selected drugs. And this is what is proven here. So in general, the activity of SYK inhibitor, MALT1 inhibitor, the pan-PI3 kinase inhibitor, or pan-PKC inhibitor is synergistic in general when you pre-treat or you weaken previously the whole system using PUT71 or HSP90 inhibitor. And the same happened with ibrutinib. Ibrutinib in combination was the best strategy, so we carry it forward. Okay, here, to animal models, the same concept. Inhibiting HSP90 weakens all the other pathway and allows the ibrutinib to work much better. We prove in animal model as well. This is our two different models of ABC, DLBCLs. You can see, compared to vehicle, ibrutinib is good. The PUT71, the HSP90 inhibitor, is much better because it inhibits many pathways. But now the combination almost eradicates the tumors in these patients. So this is a very feasible therapeutic strategy to use in ABC, DLBCL patients because both HSP90 inhibitors and ibrutinib are obviously in the clinic. Another example for GCB type of tumor comes from the idea that EZH2 is another of the supporting network that maintains the activity of BCL2 and BCL6 in the GCB DLBCL cell types. So this is with it's coming. So the inhibition of EZH2 with BCL2, with, here with ovatoclax or ABT737, seems to be synergistic in GCB DLBCL cell lines and also in animal models. And as well, the combination of EZH2 inhibitors with BCL6 inhibitors, we use FX1. So it's coming, okay. FX1 here, So this is vehicle, ECH2 inhibitor, FX1 inhibitor, and the combination almost eradicating the tumors on the GCB cell types, two different GCB cell types. So this is another example of this, awakening the network, the supporting network, and then targeting with more selective agents for the oncogenic pathways. Okay. And something else that is supporting this network is the oncogenes need to be expressed. And it is something obvious, but is, sometimes we not acknowledge this mechanism that leads to the oncogene expression. Either in expression, double expressor, triple expressor, double hit, triple hit, the proteins must be expressed, MYC, BCL6, BCL2, and FKB. And the process starts in the chromatin in the nucleus. The RNA is being produced. It has to be transported, exported from the nucleus to the cytoplasm using the nuclear pore. And then the protein is translated, the RNA is translated into protein, and the protein is folded. So this mechanism, okay. So this mechanism to produce oncogenes is different than producing a protein that is not an oncogene, for example, an actin. And this is very good because now we have a totally mechanism to produce oncogene that can be potentially targeted independently of the housekeeping proteins. So it's not going to be toxic for other cells that do not express or don't need to express oncogenes like normal cells, right? Things coming. Okay. So all this pathway for oncogene production is supported by a network of these proteins that I mentioned before. For example, BRD2 and BRD4, CDK7 at the transcriptional level, and then other proteins in the nuclear pore. In the nuclear pore that transport oncogenic RNAs is specific for these RNAs. I can be targeted as well. And the translation, the production of a protein that is an oncogene is different than producing a protein that is not an oncogene. So it can be also specifically targeted. And I'm showing here different drugs that can be used to specifically target the production of these oncogenes. I give you a few examples, if this works. Okay, here. So for example, using a CDK7 THC1, the THC1 inhibitors of CDK7 affect the region in the chromatin called superenhancers and decrease the production of MYC protein. So you can see vehicle and treated with THC1 tumors, the MYC protein decreases largely. Similarly, okay. Using this drug silvestrol that affects the production of oncogenes, we can decrease the protein of MYC, for example. Here you can compare. So silvestrol administered, other ACH2 simultaneously, right? Cyclin D3, BCL2. So all these oncogenes disappear pretty much from the cell treated with silvestrol drug here, but not the proteins that they are housekeeping, right? Actin, tubulin, GAPDH, are here. So this indicates that the drug, the mechanisms are totally different, and we can target it. This is not in B-cell lymphoma. This is a work by Guido Vendelab, but I think it's a good drug that we can use on lymphoma as well. We specifically are interested in how the RNAs, oncogenic RNA, are transported into the cytoplasm. They use a nuclear pore that is very particular because it has a very particular composition of proteins, and these proteins can be targeted. So we are studying how we can target the nuclear export of oncogenes by ribavirin, which is an antiviral drug that is repurposed as a EF4E inhibitor, and also selinexor or leptomycin as well, that are inhibitor of XPO1, the exporting one that makes these RNAs to be exporting from the nucleus. To investigate all the targets that they are exported through this mechanism. So we check in of the RNAs that are associated to EF4E. There are many, many RNAs that use this specific nuclear pore, including all that code for the B-cell receptor signaling here, and many other pathways as well, and all the network that is associated to MYC, so MYC and MYC network, BCL6 and BCL2 are exported using EF4E. Therefore, if you inhibit EF4E using ribavirin, an antiviral drug, you see a decrease in the expression of BCL6, MYC, and BCL2 at the same time in different cell lines, including double hit and triple hit cell lines. This justifies and this explains somehow what patients, and especially patients with double hit or triple hit disease, it has very high levels of EF4E because they need this nuclear pore. This needs a particular system to maintain the expression, the simultaneous expression of these oncogenes. So in general, this patient has very high expression of E4E and XPO1. This is an example of a patient that have a xenograft model, similar to what David showed before. In this case, this patient has a triple hit disease. It was very refractory to the treatment. We generate a PDX, Churchill and Girami help here, so you can compare the patient expression of these genes and E4E and in the PDX in the mice, right? So we tested with all these drugs in the animals, and it was totally refractory, like the patient, so that we consider a good model. And then when we test the triple hit PDX with ribavirin, there is an anti-lymphoma effect, which is pretty remarkable for a drug which is an antiviral drug. And the same happened when we tested this triple hit PDX with Selinexor, or the XPO1 inhibitor. So it's targeting directly one protein and one pathway in the cells, made the tumor almost gone in a very highly refractory disease. So this is the mechanism that we are targeting here, and we said, well, because HSP90 supports the translation of these particulars RNA, so maybe we can even deeper decrease expression of these networks by combining an HSP90 inhibitor with ribavirin and EF4E inhibitor. So we went back to the animal models and we did this combination. Okay. And so you can see here we use a triple hit, double hit, and the PDX from before as model for in vivo administration. Ribavirin has some effect, I showed before, PH71, the HSP90 inhibitor as well, but the combination always results in a higher anti-lymphoma effect. So I think that this combination is also extremely attractive to be used in the clinic, and specifically for patients with double hit or triple hit, very chemorefractory disease, because they're the ones that are more dependent on EF4E and XPO1 activity. So to conclude, new therapeutic strategies that are actively explored in B-cell lymphoma include combinatorial treatments to increase the effect of immune checkpoint inhibitors, a huge challenge for the preclinical model, because sometimes we use immunocompromised mice or where 2D systems are not up to this task. So we had to create new models to test this. The idea is to create a more favorable microenvironment or increasing antigen presentation. Another strategy to target big compartment therapeutics, the lymphoma cell and the microenvironment. So try to break the communication, the bidirectional communication there. And the combination of broad and restricted targeted agents I showed before with HSP90, Brutinib, and so on. So that's it.